Thanks everyone for joining us today. Welcome to our monthly meeting of the New York City Neighborhood Naturalist Group. My name is Lydia Paradiso, and I'm a doctoral student at the New York Botanical Garden and a graduate fellow with the New York City EcoFlora project. The New York Botanical Garden created the New York City EcoFlora project in 2017 with a grant from the Institute of Museum and Library Services. Building on the garden's historical strength in collections with emerging opportunities in digital technology, the EcoFlora is a community science project to document the wild flora of New York City. To date, nearly 25,000 observers have made over 700,000 observations of plants and their biotic relationships in the city. You can explore these observations on our project page in iNaturalist, which I'll link in the chat in a second. Each month, we also sponsor an EcoQuest challenge, which encourages New Yorkers to observe a particular species or a genus and their partners and post those on iNaturalist. And our EcoQuest for April is entitled Map Maple. And I've been seeing lots and lots of maples in flower and in fruit right now. So it's been really fun for me to kind of see how many different species I can find. So here's a link to the iNaturalist project page and to that EcoQuest challenge for this month. So our speaker this evening is Dr. Joshua Cheng, who is a professor in the Department of Earth and Environmental Sciences at Brooklyn College. He's also the director of the Environmental Sciences Analytical Center and also is a faculty member for the CUNY Graduate Center. There's a lot of other things, and the ASRC, the Advanced Science Research Center, and also the Macaulay Honors College. He's an associate editor for the Journal of Environmental Quality, as well as a member of the many projects, including the Healthy Soils, Healthy Communities Project, the Legacy Lead Coalition, the Research Committee for WRF Biosolids Research, and the Board of Trustees for the Mid-Atlantic Biosolids Association. Our talk tonight will showcase research findings from the Urban Soils Lab at Brooklyn College on a wide range of topics relevant to urban soils in New York City, including soil contamination and urban agriculture, soil survey, green infrastructure, tree planting, and waste management. Please feel free to enter any questions or comments you have into the chat during the talk or after the talk. And at the conclusion, we'll go through as many of the questions as we are able. If you're unable to stay for the whole talk or you want to share this talk with others, we will have a recording posted on our YouTube page where you also can find recordings of our previous talks. And here's the link for that. A quick plug to just also mention, this upcoming weekend is the City Nature Challenge, which is international scale BioBlitz. To make as many observations of plants, animals, insects, other organisms in our city over the weekend from Friday to Sunday. And there are a lot of different events happening throughout all 5 boroughs and beyond. I'll throw a link into that in a little while with some of those events if you're looking for something to do this weekend. But for now, I will turn over to Dr. Cheng.
Thank you, Lydia, for the opportunity. Let me just to get my slides. Can you guys see my screen?
Yes.
Great, thank you. So I see, thanks for all the interest and coming here after your work. I see actually several familiar names there. So I guess some of you probably are familiar with the work that we do, and some of you probably are less so. So today I'm going to talk a little bit about, give you a little bit very brief introduction about the soils in New York City, and also, maybe some of the work that we do from the Urban Soils Lab, and mainly on soil contamination and what it means for the urban agriculture and for public health. Let me see, how do I move forward? Oh. So as Lydia mentioned, actually I'm a professor at the Brooklyn College. I came to the college in 2006. And I actually didn't start my soil research work right away. spent a few years trying to find something to do. I was trained as a geochemist actually, so didn't really have much of a soil knowledge. And in 2009, we had a international conference on urban soils. Actually, the host was Grad Center. I was called on to as one of the co-organizers, actually, Grad Center. So we got a chance to meet a bunch of these urban soil scientists from all over the world, the best ones, and a lot of them from Europe, which started their urban soils research in 1980s, 20 years before the US does. So we kind of like started to talk about it, and then we started to say, who is studying urban soils in New York City? And we looked around, nobody is. So that's where we thought, oh, pretty interesting. Let's do some analysis of the soil to see, what's in there. And we found quite a bit of contamination issues. That's a pretty big news at the time, and a lot of interest from people, from agencies, from the news media, those kind of things. So that's where it all started. So we had started to put together the urban soils lab, and then and started to do some research, especially collaborating with the USDA NRCS, Natural Resources Conservation Service, a group of people who was doing urban soil survey. And by the way, New York City has the best, the most pioneering, the most leading urban soil survey program in the world, in the country. There's not many cities actually in the US has been, comprehensively surveyed, and we have the best program actually here, thanks to several of these leading soil scientists, which also served as mentors for us, for me, for myself, my students, and for the urban soil program at Brooklyn College. My approach, from my early graduate days was really to look at the urban environmental issues like water quality, air quality, all these things, and try to find solutions which are green and sustainable. So we're not trying to use chemicals, use some things that when you actually remediate the soil, but then you create another type of contamination. So we want to use these green, sustainable approaches, which is kind of the theme we are hearing over and over now. We want to have long-term urban sustainability. You don't want to do something that can be short-term, but it's not as sustainable on the long-term. So these are my kind of like a philosophy of doing research, finding these green and sustainable approaches. So we have an urban soils lab at Brooklyn College now, pretty well established since 2009, for about 12 years now. So we offer soil testing to the public, and we do a number of different research in the lab, get some funding from various different agencies and programs to do research. And also we try to, it can offer, we have courses that teach soil sciences, and we also offer public outreach. You can actually, many of you probably have heard about us. We have these, community outreach events and coming out and to help with the community. As part of the Environmental Sciences Analytical Center, which also, when I was hired as a director to manage this center with a bunch of these high-end advanced instrumentations, including ICP-MS, transmitted electron microscopes, and then X-ray diffraction and XRF, those kind of things. The main goal is to do the environmental analysis for environmental samples from sediment, soil, water, plants, those kind of things. I think mostly, we have been focusing on soils. We haven't done much of other things, but I think if there's any interest from the community, we would like to talk to you about it. We can expand the range of analysis for you. So here's my contact information. If anything you want to reach out to me, feel free to reach out to me. You can Google search and you can find my contact information and my cell phone number as well. And one of my goal is to really talk to people who want to collaborate, people who need the information. I think that's one of my job. And we have a website. It's called testbysoil.brooklyn.edu. And also, if you are on Facebook or Instagram. We have called the handle is called a BC Urban Soils Lab. If you search that, you can find us, and we're trying to really update as often as we can. You can find a news kind of thing in there. If you and on the right side, there's several pictures about what's happening in the lab. This is me doing the X-ray fluorescence analysis, for soil samples. And then the second picture here is actually a bunch of students that we took to Russia, which is actually a busy place right now. The European Russia actually going from the Arctic Ocean all the way to Black Sea for a 3-week trip. Actually, that's one of the most memorable experiences that students had actually, in their life. Many of them really loved it. Because of the war, we couldn't have it this year, but in the future, when things start to stabilize, we'll go back there again. And then the last picture here is that we were participating in the soil kitchen events that's organized by US EPA. And I think it's getting a little bit quieter because of the COVID situation, but I think it'll pick up later. So at these events, we provide free soil screening for lead and other metals, by X-ray fluorescence analysis. And also there's public health professionals now to help to interpret the results to whoever brings the soils in. So I thought that was a very useful event. And this is actually a range of the type of research that we do in the lab. In terms of soil contamination, we actually look at the soil testing, how to test those, develop those methodologies. We also work with the Mayor's Office of Remediation on the Clean Soil Bank project, which I'm going to expand, to say a little bit more later. And we also look at the bioavailability of the lead in the soil. We're going to talk a little bit that as well. And to look at the public exposure, the health risk, and what are the mitigation strategies and remediation strategies that when you have these contaminations in garden soils. And I'll mention a little bit about phytoremediation as well, which is a very popular topic approach, and I'll talk about it later. We also work with NRCS quite a bit on the urban soil survey projects, which is to see what's out there, right? To understand what's in the soil and look at the soil development processes. These are like a basic science research, understand, the soil, how soils develop in urban areas may be different than natural areas. Looking at the microbial diversity, how to see how the bugs going in the soil. Looking at the carbon stocks, especially the blue carbons in those marshlands that in the city, we have plenty of, marshes and the fringes. So these have plenty of blue carbons. Looking at the urban tree health and soil quality. So we have been working with Parks Department and the OER, experimenting using the constructed soils for tree planting purposes. And I think we just wrapped up and try to evaluate, whether that can be a feasible type of soil that can be used for tree planting, so whether it's sustainable for the long term. And there's some other projects working with New York City DEP. We kind of was for one of the first colleges actually worked with New York City to assess these bioswales and a bunch of other green infrastructure systems and how do they function, where they function, and what are the science questions there. I think we kind of got tons of data, and I think it was quite informative in terms of for the whole GI program in the city. And then more recently, we have been working with DEP and other agencies looking at the solid waste management and how urban soils can play a big role in terms of assimilate a lot of these very useful waste. It's not called a waste, it's called a resource, really, right? A resource that can use the resource to benefit us, to help to to enrich the soil, to make the soil more useful and to support the long-term goal of the city. So this is our website. You can go to testmysoil.brooklyn.edu, and it has some useful information for soil testing. And also on the right side, if you click on if you sign up, if you subscribe here, you will receive a quarterly newsletter from us, which can give you more updates of what's going on in terms of research, outreach and different opportunities kind of stuff. So check it out if you have time. Why are people interested in urban soils, and what's happening to people's researching urban soils? Urban soils is usually known to have a very poor quality. They're usually very compacted. They usually have no organics to start with and have low nutrients to start with. They usually have higher pH because of the carbonates, from the concrete. They usually just have higher pHs to start with. So these are not like good conditions for plants. And also urban soil is known to have a lot of these called human transported or human altered materials. These are anthropogenic materials. And New York City is well known for that. We have a lot of the construction debris, coal ash, those kind of materials that was buried in the past century. And a lot of these soils, urban soils, are contaminated to some degree. In the history of urban soils, it has been viewed by most of the cities as a burden mostly because of contamination, because of all these poor quality issues. People haven't really paid much attention to it, but now I think people are starting to realize it's a really, really very essential resource, right, that we have. And looking at all these, the things that happen in the city, people are trying to make the city more sustainable. Almost everything has to go through the soil, right? Soil. It is critical for ecosystem development. It is critical for this called nature-based solutions, right? This is a buzzword you hear a lot, nature-based solutions, right? And a lot of the land restoration and remediation efforts that need a large quantity of the clean and quality soil, which we are trying to figure that out, how to get that. So if you look at the literature, that you can find, so this is actually a figure. The top figure here is the total number of publications, with urban soils in its title. You can see that, it exponentially increased actually from the turn of the century, from year 2000 to today. This increased, the number of papers just exponentially increased. And some people may argue that, the number of papers published is exponentially increased anyway for any type of papers. But if you compare the urban soil papers versus all the papers on soils, right? The percentage of papers on urban soils compared to, the soil papers, the ratio is also exponentially increased. So by 2020, it's about 1.2% of the papers about soils are actually on urban soils. So this is really pretty striking. I think if you extend it to 2030, that may be up to 5%. So that's a pretty large percentage. Again, there's a lot of interest, to looking at urban soil, study it, and try to understand how to best use it, how to best manage it. So urban soils, is related to a lot of these very hot topics that we're talking about today. Look at all these words. These are all buzzwords, right? We're talking about today, from green infrastructure to urban ag to solid waste management and to this urban soil survey, which, the soil data can serve many different sectors in the city. And then through this, we can address a lot of these issues for water quality issues. So water quality issues through the stormwater management, you're reducing the combined sewer overflow. So improve the water quality, which is, the most important motivation for the city to develop its bioswale program. Reduce the flooding issues, increase the carbon storage. How do we do that, right? You grow more trees and develop the GI programs and doing to beneficial uses compost. All these things can help, and develop the ecosystem diversity. How do we achieve that? We go through soils, improve the urban soil quality, air quality, reduce the urban heat island effect, and have better public health. Public health not only through the better air quality, water quality, but also through the engagement of physical activities in the gardening, for example, mental health, physical health, and improve the food security, improve the environmental justice to involve these people from minorities to have a secure food supply, those kind of things, and waste reduction, basically, redivert all these organic waste, for example, waste is a quote, it's a resource related right now. So to go to landfills, but to just beneficially use them to improve the soil. So a little bit, a few slides about in general, what do we have in New York City, for example, in terms of soils. First thing we think about is that what are these parent materials for soils, right? There are three, if you look at New York City, there are really only three physiographic provinces. One is called a New England upland here, and the majority is called a Triassic lowland, and then Triassic lowland, and then the majority is called Atlantic coastal plain. So as you all know, I think most of you know very well, and the whole area is really controlled by glacials, right? Glacial deposits. So various different glacial deposits, and this is glacial outwash alluvium materials, and this terminal moraine here, and also this is glacial till material. And you can see if you go from north to south, you can see the grain sizes, they do change, right? And thickness of the sediments also change. These are all the materials that is the parent material for the soils that we see here today, right? So that's called a natural part of the natural soils, right? But again, on the other side, a lot of the areas in New York City are not natural soils. So these are, anthropogenic materials that serve as parent materials for these soils in New York City. By the way, I have to say, a few slides that I show here, these were from Dr. Richard Shore. I think a lot of you know him really well. He was the state soil scientist for New Jersey and for New York State. And for many years, just retired a few years ago. I'm borrowing a few slides from him. I think he's got he's probably the most, knowledgeable person about the soils in New York City. And New York City, you guys all know the history of New York City, European settlement in early 1600s. And so the population, exponentially increased since then. There's very little room for expansion, but the population increased, but a lot of things have changed from the early days. There's only limited space here. So we have about land area, about 800 square kilometers, about 300 square miles. So in the history of New York City, there's a lot of, efforts really trying to expand the land area, right? Number one. Number two is find us places to dump the trash, basically. So a lot of these really good French marshes, right? French wetlands surrounding the city were filled with trash, basically. All sorts of different trash materials. And I heard that one of those early goals was to really try to eliminate the mosquito problem from the marshes. And at the time, people thought that was a really good idea. And so they kind of filled it and solved the two problems, right? Mosquito problem and a trash problem. And now I know that everybody looking back said, oh, I wish we didn't do that, right? They shouldn't do that. But this is what we have, okay? Sorry, so if you look at the city total, we have about 63% of the area is basically concrete, buildings and roads is covered. It's called sealed soils. And we have about 28% is called human altered, human transported soils, which is anthropogenic soil. So it's a very large percentage, okay, large percentage of the area. And naturally developed soils, ND soils, it's only less than 9%. Okay, so if you happen to go to one area, dig up the soil, more than likely you're going to find the anthropogenic soil rather than the naturally developed soils. Most of the natural soils are in parks, in some of these parks that are, urban forest areas. By the way, the percent of sealed soils, the land area that's covered by concrete actually is New York City is doing pretty good. We have about more than 35% of the open space, open soils covered by soil, which is really good compared to other cities in the country. So New York City soil survey teams, through different generations now already, I think starting from 1995, have about more than 100 pedons that has been sampled, and from by soil survey teams. And Brooklyn College, our lab team has been helping the soil survey teams, and through these cooperative projects, and through, I'm going to say a little bit words of it later, the 2 projects I've been working on, and probably assisted about 2,000 sites so far. And so there are soils naturally developed type of soil series. There's 38 of them that's mapped already. And for soils in the human altered and human transported materials, there are 32 soil series mapped. So basically, soil series is the basic, the lowest unit, the smallest unit of the soils. When you dig, it has different, separate characteristics, different one different than the other. Their soils in each series would have similar particle size, geology class, and structure, and these kind of characteristics you can see in the field. It can and you can characterize in the lab. So this is on right side here on a picture here. It's a typical soils map. You can see it's divided by lines into different areas, and within the area you have different type of soil units in here. Sorry. Okay, if you are interested in getting soil information, if you wonder, say, for example, what does the soil look like, beneath my feet, right, wherever you are, and so NRCS, USDA and NRCS have a pretty robust called a National Cooperative Soil Survey Program, and they have all the data in one database. So if you Google search Web Soil Survey, you can go to this website, and they can actually, you can go in there and see, and based on your coordinates, you can find what kind of soil is beneath you, right, below you. So, I think if you're interested, if you need any help, I'll be more than happy to walk through with you to do that. And also they have I think the video somewhere here you can watch to see how to get the data. I think that the database is kind of a little bit underused to some degree because a lot of people don't know about it. But I think, if you're interested, you should look into it, to see how to do it. But again, if you need help, let me know. There's another way to get to the soil information. This is actually called a soil web. The previous one is called a web soil, right? But there's an app called a soil web. If you have an iPhone, for example, you can search for an app called soil web. And this is actually a very extremely useful and simple, very straightforward app that you can click actually, and you can use your location information. And once you click into there, it actually gives you the soil map and the detailed soil information, wherever you are. So I found it extremely useful, and you don't really need much knowledge, prior knowledge to be able to understand that. Once you get that, some sort of descriptions, if you want to understand it a bit better, you can always reach out to us and reach out to, any of the, soil scientists or resource scientists, from NRCS to help to interpret it. So I found this one was very convenient, actually, wherever you are. Of course, if you're interested. So next, I'm going to talk a little bit about the 2 interesting cooperative projects that we had worked with USDA Natural Resources Conservation Service, the Soil Survey Program. And the first one was actually really trying to understand the soil microbial diversity in urban environments. And at the time of 2013 and 14, there wasn't really much research in terms of looking at the microbes in the urban soils. And we know urban soils have a lot of these, artifacts, fabrics, those kind of things, but we didn't really know, what they do to the microbes. So you can see that, we were, in the field and digging those pits with, with students, with soil scientists from USDA really try to taking the samples, getting to the lab to do these, chemistry and microbiology, those kind of things. Was a 2-year project. You can see that we took samples from various places in the city, and they represent different soil series, different type of soils. Some are anthropogenic, some are natural. So we want to try to compare them. So one of the very interesting things about very interesting finding, I think it's pretty, to our surprise, is that there's a lot of bacterial diversity in these called human altered and human transported materials. You can see that on the right side here, these are the anthropogenic soils, the human altered, human transported soils. On the left side here, these are naturally developed soils. So there is called a Shannon diversity index. So the index is higher actually in general for anthropogenic soils and natural soils. That was really pretty surprising to us. We thought that these kind of waste material, right? Fly ash, coal ash, these construction debris, they wouldn't support much, bacterial activity, but they do actually. They have a lot of, microbial activities there. They also have pretty high abundance of these things. So many people think that if you have more bacterial activity, you seem to be, soil seems to be more vibrant, more living, right? So more healthy. That may not be the case healthy, but they definitely have a lot of these different type of bacteria and high abundance of them. And what does that mean to support the plants and other things? I think we need to know more about it. But in terms of bacteria itself, it's very abundant. It's really promising. So I think there's a lot of things that we don't understand yet, right? This is actually something, that is beyond our, I think it's pretty surprising to us. I think, and after this project and many other people actually from other parts of the country or the world, they also, find a similar findings. I think it's pretty, pretty interesting, and people are starting to discuss more about this. And the other project, the ongoing project now is we started in 2019 and it's still ongoing within RCS, is really to try to develop new methods, soil survey methods for urban areas. And traditionally the survey, for urban areas is go to some place, dig a pit, and then try to describe it, and then try to take samples, get to the lab to analyze them. It's very labor intensive, it's very expensive, and we were really trying to think about, are there better ways to do it? So one of the ideas is to use those called geophysical tools, called a ground penetrating radar and EMI, these kind of like geophysical tools. You can kind of scan it, right? Scan it at the surface without digging any pit. And hopefully that can give you some information, can be reflective of what you can find from, from down below. So as you know, urban areas, a lot of places, access is very restricted. You cannot dig pits everywhere. You have to get a permits to do those. And all these things take time, and it may not be appropriate to dig pits in some places. So if we can get this thing to work and to see what kind of information it can offer us, I think we can use this, get some useful information for majority of the areas. It can also help us to expand the areas that we can survey. For example, right now we only have about 130 pits dug. So we try to interpret everything based on these 130 pits, which is actually, pretty, low resolution. We want to be able to know more about everywhere in the city, right? Very high resolution. So maybe the GPR and EMI can help us to achieve that. We also try to work with the Rutgers University and the University of California Riverside scientists, look at some other, things, the hydraulic properties, which is a very important, in terms of how to support the use of the soil, stormwater capture and septic tanks, those kind of things. They are relying on hydraulic properties. So we want to understand, and flooding, of course, issues. So want to understand it better. We also try to look at the carbon capacity of carbon capture, carbon storage for these urban soils. want to look at the high resolution geochemical processes in these urban soils and taking advantage of these newer instrumentations at the UC Riverside. So it's an ongoing project. And you can see some pictures. Last week we were in the field, actually, a lot of people from various universities and NRCS soil scientists together digging these pits and doing the survey with instrumentation, and then also dig the pit, so to be able to compare, the type of data we get from various different approaches, to see how they match with each other, how to interpret in the future with these geophysical tools. So you can see that this is a pile of coal ash material, and this is from the Greenbelt Recreation Center in Staten Island. This is, I think, a Lenape playground, also in Staten Island. So these are more, I think, some of these are more naturally derived soils. Some of these are more, coal ash, the coal ash material. You can see this is on the right side here. This is actually a picture of the monoliths just that we were trying to make. When you go down from the surface to the deep depths, you can see the different, buried coal ash materials covered by a very thin, soil, organic soil at the surface. Okay, so I think I'm going to spend some time talking about one of the, most interesting issues, the most prominent issue in the city, which is the contamination of urban soils by lead. And you hear a lot about the lead issue in the news, every once in a while, and people, this is a news report from WNYC radio last year, lead in the land, and talking about the lead in the gardens, and also, lead in some nearby some smelters, and there's two articles actually a few years ago in 2015 from New York Post talking about lead and arsenic contamination in one of the gardens in Brooklyn. And the message was very negative, saying that you might get cancer by growing stuff in the garden, and you shouldn't get close to it. Those kind of things was pretty bad publicity at the time. I think it was really a big thing. I think there's quite a bit of work has been done, probably quite a bit of public outreach has been done around that, but I think there's still a lot of significant confusion as to what the lead really, what is the risk, and should I garden or not, at what levels can I garden, at what levels I shouldn't be gardening, and what can I do basically to minimize the risk, the gardening in the city. So we did quite a bit of work actually starting, our research from 2009 from the very beginning was really trying to understand the nature of the lead contamination, especially in gardens, and how widespread it is and how high the levels are. So you can see on the right side here is a map that we published in 2015. You can see some of you might have seen this before. And each dot here is actually a garden, and these data are based on the soil samples received at in urban soil lab in Brooklyn College, and then we plot them based on the levels of lead in there. The red is above 400 parts per million, yellow is 150 to 400 parts per million, and green is 1 to 149 parts per million, and those. For some zip code areas, there's not enough. There's only less than 5 samples, so we did not plot it. But for these places where we have more than 5 samples, and we took the median, and plotted the color for the zip code based on that median value. So if it's red, which means that the median of that 5 samples or more is more than 400 parts per million. For the 400 parts per million is actually the EPA is kind of like a sort of general threshold for for their advices, basically but it's not like enforceable standards in any way. but we also use 400 as a kind of like a threshold, general threshold. So you can see in northern Brooklyn area, and mostly northern Brooklyn and part of Queens, and also part of that Harlem area, they have pretty high lead levels actually. The median value is more than 400 parts per million. And if you go, this is the oldest part of the city, the center of the city, you have a lot of traffic, right? A lot of traffic congestions, old buildings, those kind of things, and a lot of industrial activities here. So it's not surprising they have high lead levels, in general in these whole areas. If you go, away from the center of the city to southern Brooklyn, to Queens, and to northern Manhattan and Bronx, so usually things get a little bit better and at much lower levels. But again, we found that it's very interesting that is the lead levels actually are very heterogeneous, even at very small levels. So one example is that within one single garden, some spots that you have maybe have low lead, but some other spots may have, high lead, which can be like 1 magnitude higher than actually the other spot. So it's extremely spatially highly variable even within one garden. If your neighbor has low lead, it does not mean that your garden will be safe. Okay, so just keep that in mind. It's just very, very heterogeneous. So we found that almost all the soil in the city, is to some degree contaminated by lead. And the rural soil tends to have less than 20 parts per million of lead, but most of the soils, I would say 99% of the soils in the city you can find is more than that. So it's not surprising. It's not New York City, only New York City, but it's all everywhere in the world. Cities have that. The older the city is, the bigger city is, usually the bigger the problem. It can be 10 to 1,000 times higher than background levels, which is 20 parts per million background, then 10 to 1,000 times higher can be. And many samples exceed thresholds of 400 parts per million. In our findings published, we found that about 50% of the samples actually exceeded 400 parts per million. These are all samples from the gardens, okay, gardens. It does not include samples from other places, other places. And it's especially highly variable, and even in small area. And the exposure risk can also be variable, depending on what you do with the soil, how much time you spend with the soil, and whether you have a good habit actually to wash hands, those kind of things. So it varies to some degree about your risk. We also found a very interesting fact is that community gardens tend to be much less contaminated than the home gardens. So these two figures on the left side is community garden, on the right side is home garden. The x-axis is lead and y-axis is arsenic, right? You can see, if you're in this quadrant here, the lead is low and arsenic is low, right? Both are low. And if you are in these two quadrants, either arsenic or lead are high. And then if you're in this quadrant, both lead and arsenic are high. So if you look at the community gardens, most of the data points are in this first quadrant here. So they are pretty low, right? Most of these samples are pretty low. But if you look at the home gardens, most of the samples are in this quadrant here. So which means that lead is very high. So home garden tends to have much higher lead than community gardens, high chances of higher lead. And the other thing is that these colors, the red color versus black color. So red color means that one of the other contaminants besides lead and arsenic are exceeding limits. So you can see that the home garden is dominated by these red dots, right? Which means that at least one of the other contaminants are also exceeding limits. So home garden is not only contaminated by lead and arsenic, but also by some other contaminants that are in the soil. So home garden is a bigger problem than the community garden. I know that the community garden have more people, involved, right? Home garden tends to each garden have smaller number of people getting involved, but the home gardens tend to be more contaminated. And also I think home gardens have much less resources than community garden because the information wise it does not flow, right? Community garden tends to have the green thumb gardens, for example, they have information from the green thumb resources, support from green thumb, for example, can be available. But home garden tends to be relatively isolated. I think that could be something that we really want to look at. One interpretation of that, we think why community gardens seem to be a little bit better in terms of the contamination. We think that community gardens has been better managed, because I think over time the new soils has been brought in, compost has been added, those kind of things. So over time, it's really diluting the lead in the soil. So I think that could be the main reasons. Home gardens, usually the case is that people suddenly find that, oh, my neighbor is doing something, so why can't I do it? So usually the plot usually hasn't been managed or doing anything for several decades or years. So left tons of contamination there. There's no remediation, nothing happened to it. So you have high chances of finding lead contamination and other contamination in that home garden soils. See. Another study really trying to understand how the soil lead is connecting to the blood lead. I know that some of the people in the city does not consider soil lead as a big problem. I think we had some conversation with DOH, and I think there's still quite a bit of doubters as to say how soil lead is, don't think soil lead is a big problem. But this study is really showing, on the left side is lead concentration in the parks in Brooklyn. Okay, in Brooklyn, there's I think 1,500 samples collected from various different parks of different sizes. And on the right side is actually the blood lead levels. Actually, I think in Brooklyn, you can see some similarities actually how blood lead levels is kind of correlated to the soil lead, but it's not exactly the same. Obviously, there are various different factors contributing to blood lead, and soil seems to be statistically seems to be one of those very important contributors actually to the blood lead. I know there's people are still doing research, but this is some evidences to support that kind of conclusion, right? So one of the things I think from research also try to communicate information correctly is that the soil has lead in there, but when the soil is ingested into human body, especially for kids, right? When they play, they have dust and the dirt, they put fingers into the mouth, and then once the soil particles goes through the human body, only some of the lead is absorbed and goes into bloodstream. Not all the lead lead actually goes to the bloodstream. So that percentage of lead that the total lead goes to the bloodstream is called bioavailability. So this is a very important parameter, in terms of lead risk, the risk of lead. So we use some kind of like chemical leaching of the soil and to kind of simulate how much lead is bioavailable to human, and then this is called bioaccessibility. So, in one of the studies, we found that actually the bioaccessibility of lead actually varies a lot from sample to sample, which is actually kind of different than, what you'll find for the rural soils. Rural soils, if you go to some places, it tends to have very similar bioaccessibility. The percentage of the lead that can be uptake goes to bloodstream is very similar. But in urban, it's just so complicated. Soils in urban areas are just so heterogeneous in terms of composition. So bioaccessibility is very different. That gives us, more complexity as to say, how do you evaluate the risk of, risk from the lead in the urban soils, right? That's another compounding factor. So, one of the things that we want to think about it to do is to, maybe we should not be looking at the total amount of lead in the soil. We should be looking at the amount of lead that is bioaccessible, because that is the portion that is posing the health risk. So that is how motivated us to develop called a soil testing kit, the field kit, which is not relying on any instruments or X-ray fluorescence, portable XRF instruments to measure total lead. So this one is actually developing a very low cost field methods with some scientists from Columbia University. Some of you probably know very well and worked with them. And then the low cost method to be able to determine the bioaccessible lead in the field with reasonable cost, right? Very low, a couple of dollars basically to do each sample. And so this was published a few years ago. I don't know what the status is now as to how many community people, gardeners actually are using it. If you are interested, of course, contact us, contact the folks at the Columbia. I think we should try to collaborate and try to do more about this. So right now, majority of the testing is done through the X-ray fluorescence, screening. We call it screening methods. I think it's very cost effective and it's available to you through our labs or through these outreach events. You can get a free screening for soils, which both of these I think are really important and necessary. Before you do anything, just make sure that you get your soil screening before you do anything. And I think it's not surprising to most of you as well is that we found that the vegetables that you grow in the gardens, the root veggies is not really suggested, advised to do because they can contain high levels of, lead and other contaminants. It's the fact is that it's in the soil, all the time, and then it's in contact with contaminated soils. It tends to have high levels of lead and other contaminants. And leafy greens also tends to have a lot more contaminants in there as well. And the fruit, usually have much less, and it's kind of safe to grow. That's what we want to try to advise people, especially tomatoes, for example. We haven't really found much of contamination at all, for tomatoes, even if you grow, tomatoes in a pretty, with decent amount of lead, but several hundred parts per million, and the tomatoes seem to be safe. So we want to, to encourage people to grow more tomatoes and less potatoes, right? Less potatoes, and definitely root veggies needs to, to be discouraged, especially if you have a contaminated soil. So one of the very interesting things, this may be a little bit technical, very interesting phenomena. We found that the levels of lead and aluminum actually is always very well correlated in these vegetable samples. And we did a bunch of these on radish, on lettuce, for example. So a lot of samples we analyzed, we always found this really good correlation, which made us think what's going on, right? So we all know that actually plants do not really take up aluminum because they don't need it. It's not an essential nutrient. So especially under neutral conditions, and they don't uptake aluminum, but where the aluminum come from in the plants, right? In the vegetables. So they actually come from the soil particles. So it can come from the splash, it can come from the the dirt from the particles, suspended particles in the air. They can go onto the leaves, they can go to the skins of these veggies. So these are soil particles. So the correlation, I think, really tells that most of the lead in here probably is coming from the soil particles, right? It's not really from the uptake. So this is not only one veggies, it's actually pretty commonly seen from many different veggies, right? So, and more recently working with Cornell and one of my former PhD students, we designed a really good, well-designed experiment to try to examine this process, whether the lead in the veggies actually are dominated by soil particles. So there's 3 different treatments. This was done both in Brooklyn and also in Ithaca, where Cornell University is. And there's 3 different treatments. And this is a bare soil. You grow lettuce in there. And so this one potentially can have some uptake if there is some, and can have splash during the rain event, can have splash, and then you can have some aerial deposition of the soil particles onto the leaves. So we are looking at 3 different components there, right? And for bare soil, this would have 3 different components in there. But then there's a hoop house, it's covered here. And so there's no rain coming in. All the watering is through the manual watering, but the sunlight can come in, right? It can grow. So in the hoop house, the lettuce, they only have uptake. It will not have splash, will not have air deposition, only have uptake, right? So, and then another treatment is that you have the soil covered with mulch. In this case here, you can have uptake, but there's no splash because rain comes down, go to the mulch, the soil is not splashed, right? But there's possibility of aerial deposition of soil particles onto the leaves. So this way, so you look at 3 different situations here, right? You can separate different components from uptake, from deposition, and from the splash. So here's the results. Okay, very exciting results. And this paper actually is just published. If you're looking at, you grow these lettuce in these high lettuce soil, about I think 2,000 parts per million of lettuce soil. This is grown in Osaka, and this is New York City. You can see the 3 different components: splash, deposition, and uptake. So which one has the highest percentage? The splash. Okay, in this case here, New York City also splash. So it is very clear that the majority of the lead actually in the lettuce is from the splash. Okay, splash. This is really, really important. And the second important component in New York City is from the deposition, of suspended soil particles in the air onto the leaves, which contributes to the lead in the lettuce. The uptake in both cases are pretty small percent, less than 10%. So really that confirms our suspicion that the uptake of lead is really, really minimal, right, in the case of lettuce. And this is actually pretty commonly seen for other veggies as well. So uptake is the least of your concern. The concern is the splash and deposition, okay, especially in New York City for high lead soils. So that's not number 1 question, number 1 conclusion. The second conclusion is that if you compare the veggies grow in low lead soil versus the high lead soil, you can see if you grow in the low lead soil, you have very low lettuce, lead in lettuce. So obviously, if you can have clean soil, you have, with low lead, you want to grow grow lettuce in the clean soil rather than dirty soil, right? If you have the option, right? But if you don't have the option, for example, then what you want trying to do is to minimize the splash and minimize the deposition, right? Okay, I think that's a very important point. I don't think people have really, really emphasized and found out in the past. This is one of the most exciting research findings. And the other findings is also very exciting with the same group of researchers. And we did actually try to wash the lettuce to see how effective that is. Actually, washing lettuce can reduce the amount of lead in the lettuce, right? Amount of lead in the lettuce. So this is unwashed, right? Unwashed, and this is washed with water. This is soak in the water and then wash it, and this is with some other fancier commercial solutions to wash the lettuce. You can see that as long as you wash it, okay, as long as you wash it, you can see the levels of lettuce, the lead in the lettuce actually are below the threshold of 0.3 milligram per gram, threshold. It's a European Commission standard. It's United States does not have a standard actually. Yeah. So washing is important, right? Even just with a water, just a tap water wash, you can get rid of majority of the lead on the lettuce. So that one, on one hand, it tells us that the lead is actually coming with particles. Most of these are loosely associated with the surface of the lettuce. You can wash it away, right? And the amount of that is actually trapped inside the leaf, either by uptake or by during its growth and kind of trapped in there, can't be washed off, is very minimal. So that's conclusion number 1. You need to wash it. Question number 2 is that it's a water, waterings, just wash with tap water is as effective as other, approaches. You can get a little bit of less, less, but not going to help that much, right? So, I think you don't really have to go to stores, buy these fancier solutions to wash, which has, some other uncertainties, we don't know what's going on in these commercial solutions, right? So washing with tap water is really, highly, advisable practice you need to do before you eat anything when you grow stuff in somewhat contaminated soils, right? Okay, let me see. So what does it mean for mitigation, for whatever you need to do to reduce your risk? I think there's a couple of advices. One is, first of all, if you don't know whatever you have in your soil, you need to get it screened to see how much lead is there. Maybe there's some other metals in there as well. It's highly advised to know what you have first before you grow anything, right? And two is you want to try to grow in clean soil as possible, but don't be scared if you have, soils with lead levels below 400 parts per million. I think you should be able to grow your veggies in there without much fear, but you just have to do the right thing, right? Be smart how to grow it, how to keep it safe, right? And if it's above 400 parts per million, my advice would be to build some raised beds with clean soil. There's great research from the Office of Intermediation. They can provide the constructed soils. I'm going to talk a little bit later, and for free, actually. So periodically, we send out the newsletter information about the availability of these clean soils. So if you sign up, you'll get the information. Also, be smart. Choose these, fruit veggies instead of root veggies. Also, I know that the leafy greens tend to be very popular, but just make sure that you do the right thing. And also makes doing common sense practices, washing them, peeling them to wash. I think it all helps to reduce the risk you will have from the lead contamination. A lot of these practices that has been using from in farms, especially in farms, and from encouraged by the NRCS, Natural Resource Conservation Service, for example, using mulch cover, for example, right, and that can reduce the dust, right, and then use a drip irrigation to keep the soil moist, and add amendments too. Of course, these can all contribute to the weed control and contributing to water conservation. All these are good practices, and very interesting enough that they can also help you to reduce the risk from the soil contamination, right? To do that, right? It can help. So, and also there's some conservation tillage, which means that you don't till every year, and it can reduce the dust as well. Basically, that can greatly help you to reduce the risk from the contaminated soil, right? All these things you can do. So I want to really bring to your attention called a Clean Soil Bank program, which is from the Mayor's Office of Environmental Remediation. They have been providing soils to many different projects in the city, and I think they have abundant supply of these clean sediments, beautiful, clean glacial sediments. I think they have a surplus of it from various different construction projects. And we did some research a few years ago, and mixing with these sediments with the compost material and generating a growth medium, basically deliver into the community garden, build raised beds, and then planted all sorts of different veggies and try to see what the performances are, and also monitor whether there is contamination at all. And I thought was very, very successful, and with the help of various different the Parks Department, the Brooklyn Queens Land Trust, and also Gowanus Conservancy. Everybody worked together. The results was pretty successful, I thought. And this just shows a picture of these clean sediments and pile of clean sediments. And I think the Queensboro Bridge, beneath the Queensboro Bridge, and the compost material, and my former student Sarah Eckendorf. And also this is my postdoc, actually from France, and who was actually working with these raised beds for different experimental raised beds, and to look at the performances. And the observation from this experiment was that it's a really very viable growth media for raised beds. Okay. So about the 33% of the compost, the sediment mixture, which means that one portion of compost and 2 portions of sediment, volume-wise, right? They create about equal yield as natural soils. Okay, natural soils. So if you add more compost, obviously they can actually help a little bit more in terms of the yield-wise. And I just remembered that even in December, that was a very warm winter. In December, we still have like too many supplies of these tomatoes coming out of from these plots, and we didn't have space for those fridge actually because it's a research, we couldn't give out the tomatoes to the community, but it just shows how much yield we had for that experiment. And in that 1 year period, we didn't find any evidence of contamination, and that shows that building raised beds is probably one of the better ways to keep the contamination risk to the minimal, basically, for you, right? And we have been actually, for instance, we have been trying to continue to monitor, looking at the contamination, whether it can be sustained in the long term, and the yield as well, and all these things. And so the OER, Office on Remediation, is really trying to expand into community gardens and trying to make it available to anyone who requests it. I think it's a very promising program. We're fully in support of it. The other thing I want to mention, I think it's of interest to many of you. I often hear people talk about the phytoremediation, right? Phytoremediation, and interesting enough is that phytoremediation of lead does not really work, especially in urban soils. And we did a very thorough review in 2021, and it's already published and looked at the many different case studies, in all parts of the world. And the conclusion is that, I don't think it's worthy to try it. It's not going to work. If you're expecting the plants are going to suck up a lead, and then you can, through that you can remediate the soil. I think you're going to be disappointed, right? Disappointed. There are a few publications maybe saying they added some special different amendments, and that can help to lead to plants to uptake lead, but it's a not extremely efficient way of doing it. Can be very expensive. I don't think that's the best way of doing it. So on the other hand, the approach to stabilize lead in the soil, for example, by growing plants or adding some amendments, that can be potentially effective, right? You're adding compost, for example, it can help to bind the lead, make it less available to the plants, right? So that can be an approach that might be effective to some degree, but again, the most useful, efficient way is to build raised beds if you can. And if not, then you want to add these more compost, add a new soil, for example, to dilute the lead in the soil, and also you'll improve the soil aggregation and make it more, make it more able to sustain, to keep the moisture, for example. That can reduce the dust, reduce the soil particles that can, indirectly actually reduce the risk from the soil lead. So phytoremediation is really not recommended for urban soils. I know that many people want to try it. I think it's really trying to want to demystify it. Okay, for lead. Lead is a very stable element in the soil, right? That's one of the reasons why it has, after the lead ban in 1990 or before 1990, most of the lead is still in the topsoil. It's not going anywhere. If it's very mobilized, if it can be mobilized, it would go down in the soil column. It doesn't, still in there. So it's pretty stable. It's not going to be, easily uptaken by any plants, right? So finally, I want to mention a few, one of the things to our collaborative team, and in Cornell University, there's various different researchers. I think they started actually urban, soil contamination research actually before I did. I think we have been working together since for years. There's a website called Agricultural Soil of the City. This is a good resource, very good resource for anyone doing, farms, gardens in the city. We have lots of resources there, and you can reach out to the researchers and to the extension, corner extension people, and can answer questions, for you. And also, I think the various things that we do, I think a big part of it is doing the outreach to connect to the people, in the city, to gardeners, farmers, and also, anyone who are interested in soil information and soil needs. Let's talk and see what you need and how we can help you. I think that's one of the main goals of this whole team. And I think quite a few years, best management practices for healthy gardening was developed. I think many of you probably have seen this already, and these are good, very informative practices in several different languages, and I think would really help you to reduce contamination. Again, I have to say, even if the soil is somewhat contaminated with lead, I think you shouldn't worry too much. As long as you do the right thing, your risk is pretty minimal. Also, I want to thank a lot of people and from various different agencies, colleges that really have worked with over the years. And that's, a lot of these research data can't be, achieved without them. There's a long list of the people from NRCS, and I probably missed a few, helped us, along the way. We want to add to this list. If any of you are interested in talking, discussion, potential collaborations, let us know. Let's talk. Finally, a few pictures of our students. I guess, as a public education, institution, we always put the student at the center of our focus, get them involved in not only in the classroom, but also in their real world experiences in the field. These are the student teams that the soil judging team is with. We took to Ohio and to Maryland, and they participated in the competitions, among colleges. And we were actually the only urban college team, and many kids have never seen, soils actually in their life. Well, they have seen it, but never really had experience with it. And when they get to the fast farm field, they were so eye-opening experiences for them. And most of them, I think all of them went to a pretty interesting career, and related to soils. I think we want to, to help the students to do that. I think a lot of the job opportunities in the city, even in the cities now, actually can use information, knowledge about soils, and we want to support that. And just to flip through a few pictures, this is the first class of urban soils that graduated. Many of them, actually some of them might be in the audience, and many of you are your employee now, right? So you probably recognize these faces. And students go to the green infrastructure site that we ran a few years ago. Students on the 5-borough green roof tour. And there's one student actually in Salema actually a few years ago, and we were growing carrots in New Jersey. This is a pretty tiny carrot, but it's just shows the point. And then the picture of students actually doing some farming actually in Duke Farm in New Jersey, and some experimental plots and being set up here. I think some of you probably recognize they found job within the city agencies. Some of them actually go to the graduate school later. And finally, another student actually who graduated from our department and did a summer internship in Russia, and she was digging soils, and we have been keeping contact since then all the time. She's so happy she found a career actually in soils. Anyways, that's all I have. I welcome any questions. Sorry, I probably took too long. My presentation a bit too long, I guess. Any question? Let's if we can't cover all the questions today, feel free to reach out to me, and I'm always positioned to chat with you.
Great. Thank you so much, Dr. Cheng. That covered a lot of different things, and I think that soil science is something that is probably overlooked very often, that people even more, I know people take botany for granted. I think that probably take soil even more for granted, that it's, buried beneath their feet. They don't think about it too much, but it's really important. So I know I learned a lot of things, and I hope everyone else did too. So we have a couple kind of comments and questions over here. I see that, Chris, you have your hand raised. If you want to go first with your question, and then we'll do the ones from the chat.
Okay, hopefully mine will be really quick. So one, Dr. Cheng, it is absolutely super important that you do more of this work. I think for those of us that have been community gardeners for a very long time, we've known this, and you've actually said a couple of things that for years I thought were very different. The one question I had for you was, you said that compost binds the lead, and I'm assuming some mulch binds lead. So then eventually, if it binds the lead, should we then work to, after like 2 years, get rid of that compost, or just leave it, or just keep putting more on top of it? Like, what do you do once the lead is bound with the compost?
Yeah, so what I mean by buying lead is that to make the lead less mobile. So it's like, stabilize it. So stabilize it. I think the suggestion would be adding more compost over the years. You have to do that anyway, every 2 to 3 years, I suppose, right? So the more you add compost into there, the better it aggregates the soil, have a better moisture retention capability, the nutrient retention, all these things. And also, it gives the nutrients anyway, extra nutrients anyway. So if you're continuously adding compost, you should be fine. So there was people talking about maybe, right, you're getting more and more lead in the soil, but it's not the case, because you're adding, pretty big amount of compost all the time, right? So that's a goal, right? Yeah. Anyway.
Nice. And I guess something I was thinking about that you ended up addressing is, I guess a lot of the lead is historic lead contamination from the, gasoline and all these kind of things from the past, 200 years we've been in this place, that it's,
yeah,
experiencing the effects of all the past.
Yeah, that is correct. That is actually, I probably didn't mention it. It's a majority of the lead is from the leaded gasoline and leaded paint in the old houses, and this is actually it's the same in very unfortunate. And also one of the source of lead is from the incineration. We had about 50 years of history for solid waste incineration. The fly ash went everywhere in the air and deposited. There's a study actually about 20 years ago looked at the sediment cores from Central Park Lake, and found a very interesting, the distribution of lead through the profile. So it can actually, very neat study, I have to say, looked at the lead output from various different sources. Yeah.
All right, we got another question here from Cynthia, who asked, does the pH of the soil impact the uptake, or even, I guess, the availability in general of the lead?
Yes, it does, definitely. So under neutral or near neutral conditions, there's very minimal lead uptake at all. But if you acidify it, it does, increase the potential for lead to be uptaken. Especially, when you get to lower than 5, I think but lower than 5 is not good for most of the plants anyway.
And another related question from Meredith is there any research on PFAs in biosolids in New York? And are there ways to remove those kind of things or to remediate? Yeah.
Yeah, the PFAS is the hottest topic right now. I think there's a lot of debate as to say, whether we should use biosolids for land application. I think, things like that. We did some work with GDBP, but we did not actually look at the PFAS. I think there's national, EPA has some research projects, gigantic research projects looking at that whole issue now. I'm not aware of any data for the biosolids produced in New York City.
Lots of compounds and lots of research has to be done on all these things.
Yeah, there are. I think also, there's a lot of, it's at a really early stage. I think there's methods, even analytical methods has not been fully developed for analyzing soils or biosolids for the PFAS, material. So it still has a long way to go before we fully understand. And the other thing I have to mention is that not only biosolids have PFAS, tons of other things in our daily life has PFAS. So we can't just stay focused on one thing. We have to look at the things holistically. What are the other sources of for any sorts of these called a fire resistant material? The carpets have PFAS in there, right? Carpets and anything. Anyway, there's a lot of sources of that. So just want to to look at it, things more comprehensively instead of just looking at one thing.
All right, I'm going to read another one from here, and then Chris, you'll be next. So we have a question from Carrie, not talking about contamination, going back to just talking about soils, that there was a lot of articles about the invasive jumping worms that might have had dramatic impacts on soil structure and decrease in soil organic material. And do you think that this was another case of, media hype, or is this actually a problem?
Yeah, I'm not an expert on it, but based on my understanding, it is actually a pretty serious issue, and because I think in the Van Cortlandt Park, or or in another park in Snag Harbor, we actually were in the field. We saw these activities that destroy soil structure, and then carbon. The soil carbon is basically gone, for a lot of surface soils. So it's pretty serious issue. But I'm not sure what the solution to this is, but it has a big impact on the ecosystem, definitely.
Interesting. All right, Chris, go ahead.
Dr. Cheng, it's great to see you. I don't know if you remember me from Sustainable Flatbush, but I know we visited your lab way back when, maybe even a decade ago or so. So it's nice to see you here.
Nice to see you.
The question I have is about the atmospheric deposition on really kind of two parts. One is, what are the current city sources for those? Is it just dust getting kicked up from disturbance of the soil, or are there other non-point sources like tires on roads or whatever? And then the second is that a concern for those clean soils? Like, we're bringing clean soils in and it's just going to get recontaminated.
Yeah, that's a really good question. I think, there's multiple different sources. There are, resuspension. There is the automobile exhaust. There's, a lot of these are much lower level of contamination than used to be, in the air, but still over time they can contribute. I think that's some of the things that we try to understand a little bit better. What is the magnitude of these things? But our thinking is that actually the deposition is probably, not going to cause significant impact on the soil, or in a matter of decades. So I think unless you're really like tracking the soil, say you're mixing the soil some way, because if you compare the levels in the soil versus versus what's in the air, if you do called a mass mass balance, it's going to take a while. It's a long while before it can really make any changes, observable changes, basically in the soil. So I think to answer your question, I think it's probably not going to be a big problem, in at least in a few decades.
All right, and I have one more question about kind of the research, the methods for doing like the soil pits. So these are, I notice a lot of shovels. So this is all, they're dug by hand. Like how is that? Because I know sometimes when I was a kid, we would try to dig in the backyard, and it gets really difficult after a certain point, depending on what kind of soil. And I think maybe in the city, there's maybe more soil compaction.
Right,
like
right.
well,
this is I think this is our warm up, basically. Save you a trip to the gym. So I think we're getting used to it, and it's actually not too bad, and we really kind of appreciate the opportunity to go to the field, doing some physical exercise before actually, doing description kind of stuff. So we also try to get more students involved, so which students are always very happy to help out in doing these things. But it's not too bad. Digging pits is not too bad. It's part of what we are proud of, I think, to do that. Yeah.
And how deep are they usually?
Yeah, so it depends where you are, and as deep as 1.5 meters. And most of the time we want to try to get but beyond the 1 meter, but sometimes we hit the water table, for example, or, and last week we had 2 pits actually was flooded in the pits. So we go as deep as we can, depending on where you are. Some places are easier to dig. Some places like fly ash is actually, coal ash is pretty easy to dig. But some places you have these chunks of stones. That's kind of a nightmare when you try to dig down. So anyway, yeah, it's a fun experience, I have to say. It's a fun experience. Yeah, it's not as bad as people think.
And I know that there's at NYBG that there is a soils class, and I've seen in the classroom they have some of the models where they have all the different layers.
The monoliths, yeah, we have collected a bunch of those in college, and if anybody interested, you're welcome to visit, and we can show that. We maybe in the future we can organize some kind of exhibits at the NYBG, for example. It can, we can always talk about different opportunities and possibilities. Yeah,
because again, this is, I think, something a lot of people don't think about, and especially for gardening and horticulture, it's really important to know. So so thank you again for coming and speaking with us, and thanks to everyone for being here and for your great questions. Before we go, I had mentioned this before, here's a link to some of the events happening this weekend for the City Nature Challenge. Hopefully we'll have some, I haven't looked at the weather forecast, but I'm hoping that it will be good at least one of the days, if not all of them, that we'll be able to get out and make some observations. And again, if anyone has any more questions, I know Dr. Cheng has said many times, if you reach out to him or any of these other things. And I do know also, I don't know when there's the next, if there's any planned events with the soil testing, but I do know that in the Bronx, the Bronx River Alliance has been having some community soil testing days up here. So
yeah, sign up for our newsletter. Actually, we'll be able to send this information occasionally. Yep.
Awesome. All right. So
Great.
Thanks everyone, and hopefully we'll see you again next month for our talk. And there's my cat.
Thank you. It's good timing, right? Okay. Bye-bye. Thank you.